Hello, everyone. My name is Gina Wang. I did research over the summer and will continue my research for the school year. Before I start, I would like to share a number with you. 10 trillion. It's such a big and long number, isn't it? Can you guess what this number is? It's the number of cells in your body. Now let me give you another number. 100 trillion. Can you guess what this number is? It's the number of bacteria inside your body. Can you imagine when you think there's so many cells inside your body, there's actually 10 times more bacteria inside you. And that is the reason why I wanted to study the communication between bacteria over the summer. So back to my title, LOX-I, LOX-R type quorum sensing system in K61, the current species novel, growth and AHR production in magnesium sulfur-rich brine. And now let me explain those big words in the title. Before I go into the bacteria I was studying, I would like to first introduce the plant here, taro. Taro or kalo in Hawaiian has an important historical and cultural impact. And now here's the reason why I wanted to talk about taro first. The bacteria my research focuses on, K61, a novel Dicera species that is expected to be named Dicera wulopoensis, was isolated from the Eloi at the Wulupo Heiau on the eastern edge of Kauai Nui Marsh in Kailua, Hawaii. And this is one of the places where you can find an abundance of taros. The picture here is K61 under microscope with 100 times magnification. And the picture here is K61 on glucose extract agar plates. Some relatives of K61 are plant pathogens, such as Dicera didonti. However, there is no evidence yet on whether K61 is pathogenic or symbiotic to taro. So learning more about K61 may be beneficial in protecting taro in Hawaii, and it will also add to the literature regarding species in the Dicera genus. Over the summer, we tested K61's tolerance and response to magnesium sulfate-rich brine. You might be wondering, why do we choose magnesium sulfate-rich brine? It was chosen because it has astrobiological, medical, and agricultural implications. In an astrobiological context, magnesium sulfate-rich brine was present on Mars during the Hesperian age, which was 3.7 to 2.9 billion years ago. The Asian Gale crater lake on Mars today is still in abundance of magnesium sulfate. Therefore, the bacteria's tolerance level to this brine is significant for forward contamination, which is the transport of Earth-based microbes to other planets. And it's also significant for the potential of unicellular life persisting on Mars, both in the past and in today. In a medical context, since dating back to the 17th century, magnesium sulfate in the form of Epsom salt have been used as mild diuretics and provide temporary relief of local inflammation. In a previous study, magnesium sulfate was used to treat rainbow trout with intestinal parasites. The mortality rate of rainbow trout was reduced immediately. And even now, you can still buy Epsom salt in drugstores like Lawns. In an agricultural context, magnesium sulfate may play an important role as plant fertilizer by supplementing magnesium to deficient soil. Quorum sensing system is a communication system between bacteria. LOX-I, LOX-R type quorum sensing system is one type of quorum sensing system which this study focuses on. And here is how it works. LOX-I over here is a gene. When it is transcribed, which is the process of the cell making an RNA copy of this gene's DNA, it acts like the mouth of the bacteria. It would send out a signal called acylhomoserine lactone, or AHL for short. These signals can then move freely between membranes. LOX-R is also a gene, and when it is transcribed, it acts like the ear of the bacteria. It is able to detect the signal AHL, which is sent by the mouth. And once the signal AHL accumulates to a certain threshold, the ear LOX-R will react and respond by activating the transcription of target genes. The concentration of AHL reflects the cell density, because the more cells there are, the more LOX-I mouth there will be, and the more AHLs will be produced. So if it's a low cell density, there will not be enough AHLs, and the gene transcription will not be activated. However, at high cell density, there will be a lot of AHLs, so the gene transcription will be activated. The way of communication is essential to bacterial activities, such as biofilm formation. Biofilms are the sticky, slimy things you could find on your bathtub, in your kitchen, or on any surfaces. And biofilm consists of many cells stuck together onto your surface. Slimy substances are secreted by the cell so that they're embedded inside these slimy layers. Biofilm help with bacterial survival, especially against harsh environments, such as high salinities. How quorum sensing plays a role in biofilm formation is that it allows the cell to communicate with each other and show how many of them there are. So when they reach a sufficient cell density to make biofilm, the concentration of AHLs will reflect that, and the gene transcription for biofilm formation will then be activated. So it's important to learn whether K61 has a quorum sensing system and how it works, so that we might know how to promote or inhibit the growth of it in response to future interaction between K61 and taro. That led us to our first research question. Does K61 utilize LOX-I, LOX-R type quorum sensing? To answer this question, K61 and CV026 was streaked perpendicular on agar plates. CV026 is a biosensor because violacin, which is a purple pigment, is only restored inside it when AHLs with a chain of 4 to 8 carbons in length are detected. As you can see in the picture here, the production of violacin was restored. This result indicates that K61 produces AHLs with a carbon chain from C4 to C8 in length. So yes, K61 does utilize LOX-I, LOX-R type quorum sensing. We also use a hidden Markov model and other bioinformatic tools to annotate K61's genome, and we identified some LOX-I genes and LOX-R genes. There were 1 LOX-I gene identified in K61, as shown in the table on the left. And there were 7 LOX-R-like genes identified in K61, shown in the table on the right. This also proves that our answer to the first research question is correct. K61 does utilize LOX-I, LOX-R type quorum sensing. So our next step is to find out what types of AHLs K61 produced. We extracted AHLs produced by K61 and used liquid chromatography mass spectrometry, or LCMS, to determine their types and relative amounts. Because LCMS is a targeted analytical technique, 10 AHL standards were used, as shown in the table here. Only AHLs in this table could be detected in our experiment. AHLs all have an universal base called homoserine lactone. However, their acyl chains may vary. For example, C6-oxo here is called C6 because it has 6 carbons in its chain, and the oxo indicates that C6-oxo has 2 additional oxygens in its chain when compared to C6. Among the standards, 5 types of AHLs were detected in K61: C6-oxo, C6, C7, C10, and C12. And this led us wondering how the AHLs produced by K61 will change if it is inside magnesium sulfate-rich brine. To decide which concentration of magnesium sulfate-rich brine will be used as an experimental group, K61 was grown in various concentrations of magnesium sulfate-rich brine. K61's growth was determined by measuring the change in optical density by spectrophotometer. Because the more bacteria there are, the more dense the solution will be, and the more light will be blocked. Among the concentrations tested, 0 to 10% had approximately the same optical density after 24 hours. The optical density of 20% was lower, but still enough to show growth. However, in 30%, growth was observed after approximately 1 week, and observed in 40% after around 2 weeks, and in 50% after around 3 weeks. Given the time constraints of this experiment, we decided to use 20% magnesium sulfur-rich brine solution as our experimental group. We did our experiment in triplicates. K61 was cultured in the control group, which was no or 0% magnesium sulfur-rich brine, and in the experimental group, which was 20%. The optical densities were measured at various stages in K61's life cycle to create growth curves. At 24, 48, and 72 hours, ethyl acetate was added so that 2 layers were formed. The top new transparent layer was the ethyl acetate with AHLs dissolved inside, and the orange bottom layer was cell debris. The cell debris layer was then drained out. The remaining solutions were centrifuged in smaller vacuum tubes for further sedimentation, and the orange cell debris layer was once again drained out. Then the remaining solutions were poured into a round bottom flask. Then by using a rotary evaporator, we were able to get rid of most of the ethyl acetate. So now it's just the lipids remaining, and AHL is a lipid. The type and relative amount of AHLs were then identified from the remaining lipids by LCMS. The graph here are the growth curves of the experimental group, which is in red, and the control group, which is in blue. The x-axis is time, the x-axis is time in hour, and the y-axis is the absorbance at 525 nanometers. The two groups reach its exponential phase at approximately the same time. However, for the experimental group here, it actually had more cells present in the mid-late exponential phase. Also, at around 72 hours, the control group, which was no magnesium sulfur-rich brine, reached its carrying capacity. But the experimental group, in 20% magnesium sulfur-rich brine, K61 continued growing. This shows that K61 appeared to like 20% magnesium sulfur-rich brine, which is very interesting and surprising. Here are the results of the AHL extraction. The x-axis is also time in hour, and the y-axis is the absolute peak area of AHL. In both groups, K61 produced C6-oxo and C6. These two AHL show an inverse relationship during K61's growth cycle. C6-oxo, which is in dark blue bars, will produce less as K61 nears carrying capacity. However, C6, which is in light blue bars, will produce more as K61 reaches carrying capacity. Also, in the experimental group, C7, C10, and C12 were no longer produced, but C8, C14 oxo, and C14 were produced instead. K61's abundance growth in 20% indicates that it is Epsom tolerant and might even benefit from the presence of Epsom salt. This has great significance for the forward contamination of Mars and the medical and fertilizer use of Epsom salt. Since C8, C14 oxo, and C14 were produced in the experimental but not in the control group, these AHLs could be important to K61's survival in high salinities. K61's ability to withstand magnesium sulfur-rich brine might also be a result of biofilm formation. As the pictures show here, in 20% magnesium sulfur-rich brine, there is a pink thin layer of biofilm that was formed at the air-liquid interface. And here, in the 30%, which is the bottle in the middle, you can see a really thick pink layer of biofilm that was formed. But in this picture, which is 0%, no biofilm was observed. For future research, we could generate growth curves for higher concentration of magnesium sulfate-rich brines. We could also look at the transcriptomes of K61, which is the full range of messenger RNA, to see in different phases to analyze what genes are being modulated by the presence of magnesium sulfate-rich brine solution and how the transcriptomes change over time. And we could also test out how K61 responds to other environmental conditions found on Mars, such as low pressure and anaerobic environment. We could also use internal standards during determination of AHLs. We could use more of those internal standards so that we could potentially detect more types of AHLs. Here are some advice to future K fellows. First, start early and be organized. Second, communicate actively with your mentors and ask questions when needed. My mentors helped me greatly over the summer, and I really learned a lot from them. And most important of all, remember to enjoy it and have fun. I want to give special thanks to Mr. Jack K, the K Fellowship Program, and Yolani School for sponsoring this research, and the University of Hawaii at Manoa and Donachie Lab for providing equipment and a lab for me to work in. I would like to extend my great gratitude to Ms. Christy Handel and Dr. Rebecca Prescott for mentoring my research and experiment in the lab, to Dr. Evan Chan for guiding me through the complicated paperwork and having weekly meetings with me to ensure smooth progress, and to Dr. Stuart Donachie for opening his lab's capabilities to me. I'm also grateful to Dr. Timothy O'Donnell for performing chemical analysis, to Dr. Jennifer Saito and Mr. Sean Larry for helping me and supporting my lab work, and to Mr. Steve Case and Ms. Lamami Kikina for helping me to pass the biosafety training and get my certificate. And a big thank you to Ms. Christy Handel, Dr. Rebecca Prescott, Auntie Lei, Jialu, and my parents for helping me prepare for my presentation. This research would not have been able to happen without the support and help from these individuals. Thank you so much. Here are my works cited for the literature and my works cited for the pictures. Thank you, and please feel free to ask or email me if you have any questions. Thank you.